Today we will continue talking about the nervous system. We will be beginning the next chapter titled just that. So that's chapter. Oops. Okay, there we go. The lines. Chapter 8A slash 2, which is how the nervous system works. And in this, we are going to focus on how a nerve impulse is generated and travels along a neuron. So we start off with, right, and so far we've understood that it's basically because of the distribution of ions. Distribution of ions, and primarily, right, the sodium and potassium ions across the membrane of nerve fibers. And I say nerve fibers because these can be axons or dendrons. And so axons are the ones that are moving away from the cell body, and dendrons are ones that are moving towards the cell body. And so, right, you have the different phases. You have the resting potential, then you have the action potential, and then you have the repolarization. Right, and there's other things that happen within these phases. And so let's start talking about the resting potential first. So what happens is, right, when an axon, and the reason they talk about axons is because when studying about nerve impulses, the motor neurons have been widely studied, right? So they're easier to study. So, right, but the concept applies to dendrons as well, right? So just nerve fibers in general. And so, right, when a fiber, or in this case, as they mentioned, the axon is not conducting a nerve impulse, it is said to be resting. So is resting when not conducting a nerve impulse. And so what happens is that the concentration of positive ions outside, right, the membrane is greater than, right, in the cytoplasm of the fiber or the axon. inside the cytoplasm. And so what that does is that, right, comparing the distribution of ions, it is, right, more negative on the inside. Now, how that's achieved is primarily by actively pumping sodium ions outside. So there is a sodium potassium ATPase pump. As the name suggests, this is a protein that is pumping sodium and potassium, and it uses ATP. So this is an active process, right? And it is pumping, right, sodium against the concentration gradient. It's pumping it out. Against the concentration gradient. And it's bringing the potassium ions in. So these guys are being moved inside the cell or the site, like, the axon. Okay. So when the sodium ions, right, sodium ions are pumped out, again, as I mentioned, against the concentration gradient. And so the concentration inside falls, obviously. And the thing about the membrane of the axon is that it is impermeable to sodium. So the membrane is impermeable to the sodium ions, and so they can't move back in along, they can't diffuse back in freely. So recall facilitated diffusion, right? So that can happen, but it's not happening. So it cannot move back in freely. And then, right, potassium is being actively moved in. So again, right, that is also, right, an active transport process. And let's, right, for sodium, let's say it's relatively impermeable. So it's not going to happen easily. Right, so potassium is being moved in by the pump at the same time. But the thing about potassium is that, the membrane is permeable to potassium. So, right, they tend to diffuse back out as well. But their rate of diffusion is slower than, the rate at which they're actively. So this is happening faster when they being moved in, and so there's more potassium moving in than there is diffusing out. Now you'd say, well, they're both positively charged, so how is that creating potential, a difference in charge? And that's because the pump, right, it moves 3 sodium ions out and 2 potassium ions in. And so there's a difference, right? You're losing 3 positive charges, you're bringing in 2, so overall you are losing positive charge. And if that continues long enough, then the inside, as you mentioned at the start, right, of axon becomes more negative than outside. And so the resting potential of a nerve fiber is minus 70 millivolts, right? So the difference between the outside and the inside is minus, like, 70. It's more negative by 70 millivolts inside than it is outside, right? And that is called the resting potential. And right, this case, right, is called that it is polarized. Is that clear?
Yes, sir.
Okay. So there is an illustration for all of this in the text. You can take a look at that, right? And obviously the explanations there as well. And so next we come to the action potential. And so what happens is when, a stimulus is received, right? So either the sensory neuron receives a stimulus, or, there are neurotransmitters arriving at a motor neuron. And we'll come to neurotransmitters later. In either case, what happens is, right, there is a change in the permeability of the axon membrane. And again, right, axon is being mentioned because that's what's been studied. Otherwise, it's the same for any nerve fiber that's, transmitting an impulse that's being stimulated. So it could be a dendron as well. And so in either case, what happens is that the permeability of the membrane to sodium ions changes, as the word they use, drastically. That's like a huge change. And what happens is that sodium ion channels, or sodium gates, so these are ion channels. So remember, right, channel proteins, so facilitated diffusion. These guys open up. And we know that the concentration outside is much higher than inside. And so sodium, diffuses in rapidly. So the sodium ions diffuse in rapidly because there's a steep gradient. And what that does is that very quickly uh, increases the right charge inside. So it goes from minus 70 to plus 40. And so that's a huge difference, right? And in this case, the axon is said to be depolarized. And this lasts for only 1 millisecond, right? So it doesn't last very long, but it's enough. Oh, by the like, so is a diffusion gradient plus an electrochemical gradient because of the difference in charges. So both of them. So, right, the potential difference goes from minus 70 to plus 40. And when that happens, right, an action potential is generated. Which basically means that now the fiber is going to, carry the impulse forward. Now it doesn't just happen, right? So there is a threshold. There is something called a threshold potential. And that is somewhere around minus 50 millivolts, right? So the whole point of this is if the fiber is stimulated enough, right, for the potential difference to go from minus 70 to minus 50 millivolts, right, then it will go all the way. Right, goes all the way to being depolarized. So depolarization, which is plus 40 millivolts. If it stays below the threshold, then an action potential is not generated. Right, so in this case, right, nerve impulses are sort of an all or nothing response. You don't have, oh, there's like a small action potential. There's nothing like a small action potential. It either happens or it doesn't happen. But for it to happen, it needs to push the potential difference from minus 70 to minus 50, approximately, right? So once it reaches minus 50, and they're like, okay, fine, we're good to go. And so then, it takes it all the way to plus 40 millivolts. Is that clear?
Yes, sir.
And right, the size of an action potential is always the same, right? So whatever uh, you know sensory or motor neuron or whatever stimulus it is, if it's firing an action potential, it will go up to, right, plus 30. Some is, some text say plus 30 or plus 40, so just about that. So it's always the same. Now, something that isn't given in the text, right, if the size of an action potential is always the same, then what constitutes a strong stimulus or a weak stimulus, right? So this is not showing up on my whiteboard for some reason. Oh, there you go. Pretty bad lag. Okay, so a strong versus a weak stimulus. And so I'll be brief because it's not part of the Edexcel syllabus. It's not mentioned, at least in this chapter. And so basically what they talk about, right, the reason is how many and how often in succession are the nerve fibers stimulated, right? So for a weak stimulus, right, only a few fibers are, activated. Again, right, if a fiber is activated, the value of the action potential is going to be plus 30 or plus 40 millivolts always, right? But only a few of them are activated, right? Or the frequency of the impulse is low. So it's not happening very often. Whereas for a strong stimulus, right, many of the fibers are being activated for the same thing. And or, right, so, right, they are being activated at a higher frequency. So, right, in quick succession. Let's fit that whole thing over here in quick succession. Okay. So that's basically, so an example is, right, the saltiness of a particular food item that you're consuming. And so if you feel that, well, it doesn't have a lot of salt, and so that means that the receptors in your tongue for salt are, only a few of them are being activated, and right, the frequency is low. It's right, so it's not happening like, one after the other immediately, right? And this is like an or thing. Whereas if you feel that, there's sufficient or there's like too much salt, for example, in something, it's like, oh, this is very salty. And that's because the amount of salt is activating far more nerve fibers for that particular taste uh, in your tongue. And the frequency of that action potential being generated is much higher. So it's like one after the other, right? In quick succession. Does that make sense?
Yes, sir. It does.
Okay. Now, when after an action potential has fired, right, things need to go back to normal. So after an action potential. And so, right, again, the depolarization lasts for about only 1 millisecond, but that's enough. So what happens after that? So like after that, the sodium ion channels that had opened to, let it quickly flow in, and it's only happening for 1 millisecond, so you can imagine how quickly, that diffusion is taking place. And again, right, so that also tells like how steep the gradient, the concentration and electrochemical gradient is. Let me just correct that. So that's concentration and electrochemical gradient. Okay. So, right, the sodium ion channels close. And right, the sodium is again actively pumped out. Right. And the right, temporarily the permeability of the membrane to potassium ions increases. And why is this happening? Because voltage dependent potassium ion channels open due to the repolar. So this is right, what this is called is repolarization. Once an action potential has fired, it's going to repolarize. So the inside is going to become negative again. So that's repolarization. And so the permeability increases because voltage dependent ion channels for potassium open. And so potassium ions are also diffusing out. So in this case, you're losing both sodium and potassium, and so you can like you're losing all all the positive ions. Well, again, not all as in everything that's present, but the major players. And because you're using the term diffuse, they're moving along their concentration gradient, right? So this one's not an active process for potassium as far as the ion channels are concerned, right? The pump, right, is an active process. And so and again, it's not just concentration, it's also electrochemical. So now what's happening is that the inside of the axon actually becomes a bit more negative, right? So it actually goes, instead of -70, it goes to -80. So right? So it overshoots a bit. So so -80 millivolts, which is even more negative than the resting potential, right? And so this is like an overshoot that happens during repolarization. And then, right, within a few milliseconds, the resting potential is restored, which is -70. And now, right, the axon is ready to carry another impulse. But there's like a bit of detail to that as well. So it's not going to just happen. Uh, right, what happens is after an action potential has fired and the resting potential is right, you know, being restored, there's a period called the refractory period. And so this is the time where the axon membrane is recovering after an action potential. right? And so it is also the time where the membrane is being repolarized. So, right, the very first millisecond after the action potential, the the axon cannot be stimulated at all, right? And the sodium ion channels are completely blocked. So even if a stimulus is coming in, it's not going to have any effect. And that is called the absolute refractory period. Right, so this is about the first millisecond after the action potential. And the sodium ion channels are blocked, right? They will not open. And so another action potential cannot absolutely fire. Cannot be generated. And then is the relative refractory period. And this lasts for a few milliseconds. And so during this time, the axon will only respond to a much stronger stimulus. Right? Why? Because the threshold has been raised. So that minimum change in potential difference that's required to go all the way, that's been increased. So it's not minus 50 now, it's perhaps minus 40 or minus 35 or whatever. Right? It doesn't stay that way. It's only during the relative refractory period. And so during this time, the voltage dependent potassium ion channels are still open. So only when those all of those close, and right, the so only when all the K plus ion channels close and the resting potential is fully restored, right, is the axon ready for the next action potential. And so what this does, right, the refractory period is very important. Why? Because it limits how many impulses a fiber can fire. So, and we say, oh, that's going to be small, so it limits the number of impulses in a fiber to 500 to 1000 per second. Right, and it also ensures unidirectional flow of the impulse. Unidirectional flow. Right, so it's not going both ways, it is only traveling from the receptor along the sensory neuron to the central nervous system, and then from the central nervous system there's a relay neuron between, obviously, and from the central nervous system along the motor neuron to the effector, and not the other way around, right? So the nerve impulse along the nerve fiber is only going to go in one way, right? And
Yeah.
right, that's because right, once, right, and we'll see next time why. Because once the potential as in fact the potential is passing along the fiber, the part ahead is becoming depolarized and the part behind the impulse is being repolarized. And so when the part that's being repolarized, right, and so, you know, the impulse won't be able to go back because it's in the refractory period. It's like, no, bhai, right, I need a break. And so that ensures that it only goes one way. Any questions about anything we've talked about today?
No, sir.
All right, then I'll see you next class, inshallah, and we will talk about, right, how the impulse travels along the neuron, in the sense, right, what's happening for it to move forward. We've talked about how the potential differences change through the impulse, but how does it travel along the neuron, right? So we'll talk about that. Okay.
Okay, sir.
All right. See you next time, inshallah.
Inshallah, sir. Allah Hafiz.
Allah.